let's review where we are in the central dogma, or the process by which we actually express the information that is encoded in our DNA. So there are two steps in the central dogma. The first is transcription. And in the process of transcription, we use one side of our DNA, the template side, to make a messenger RNA. Now that messenger RNA is going to code essentially the same information as our coding strand. The only difference is because we've switched over to RNA, we now have incorporated uracil instead. So this process of transcription takes a short message from our DNA and moves it outside of the nucleus, away from all the rest of the DNA so that it can be expressed. So we talked about the differences between RNA and DNA, and we talked about the coding strand and the template strand. So let's talk about the second step in this pathway, which is translation. During the process of translation, we are going to take the information contained in our RNA molecule, which is still a nucleic acid, and turn it into information that can be used in the cell. So during this process, we are going to use our RNA to make an amino acid chain. That amino acid chain, also called a polypeptide, is going to be folded and processed into a functional protein. Now that protein is what gives us our phenotype. So outlined another way, the RNA transcript that's going to be translated is called the messenger RNA. And again, messenger RNA because we're going to take the message that we need from the DNA and make some information from it. That process of translation is going to create an amino acid sequence, which is then going to be folded into a 3D structure, into a functional protein. So until we actually get that 3D folded and processed structure, our proteins have no function. That three-dimensional structure is very much related to the function of the protein. If your 3D structure is not correct, you have no function. So this idea of structure and function being very closely tied is really important in biology. And in fact, we have lots and lots of structures that have very specific functions, and proteins are no different. If we have a change way back in our DNA that produces an incorrect or different messenger RNA, we're going to get a different functioning protein. That protein might have limited function, it might have no function at all. And when we have a different protein, we can end up with different phenotypes. As a general rule of thumb, each individual gene is going to encode one polypeptide, or one amino acid chain, and therefore one protein. So this is sort of a general rule, and this was an idea put out roughly around the time that Watson and Crick were working on their structure of DNA, maybe even a little bit before. So this idea was put forth by two men named Beadle and Tatum. Now they were working with Neurospora, which is a type of mold, and a type of fungus. And they were looking at a particular variant of fungus that could not produce a particular amino acid called arginine. And they wanted to know what was involved in this whole pathway of producing arginine. So we can get amino acids from our diet, but our bodies can also synthesize them. So they wanted to find out how this amino acid was actually produced. And along the way, they came up with this one gene, one protein, or one polypeptide idea. So the idea is that in our DNA, or in Neurospora DNA, you have sections, you have genes, and each of those genes is going to encode its own specific protein. So you have, along this pathway that produces arginine, you have 3 genes that are involved. ARG4 is transcribed into messenger RNA, translated into a polypeptide, which is folded into a functioning enzyme. Now remember, an enzyme is a protein that catalyzes a metabolic reaction. So that enzyme is produced, and that enzyme acts on part of the pathway for the production of arginine. The second gene, ARG2, produces an enzyme that is used later in that step. So again, we have our gene to messenger RNA to protein. And then finally, we have a third gene, ARG1, that is involved in catalyzing the reaction of producing an enzyme that catalyzes a third reaction before we get to the production of that amino acid. Now, all of these genes are probably related to one another. They're all performing a similar type of pathway, and they're probably also located nearby on a piece of DNA. And in general, our genes are going to produce one type of polypeptide. Now, this is all well and good, and it works very nicely on paper, but in actuality, sometimes you can get more than one polypeptide from a particular gene. And a particular gene often contains information to make multiple related polypeptides that each have their own specific function. Now, don't confuse this with the 3 different genes that are shown here. These are actually different genes, and in this particular pathway, each gene produces one specific enzyme. But some genes can have some genes have the ability to code different polypeptides, and those polypeptides are made by processing them slightly differently. Overall, you can use this rule of thumb of 1 gene, 1 polypeptide, just to make things easier. And we'll talk later about how the processing can actually, how you can process a polypeptide to make different functional proteins. Further complicating this idea is the fact that some genes actually encode other types of RNA, not just messenger RNA, instead of proteins. And so in this case, you would have a gene coding for a RNA molecule rather than a protein. But the same idea would still take place. You would still have a gene coding a particular functional RNA rather than a protein. So in general, genes are going to produce polypeptides. 1 gene, 1 polypeptide, but there are some important caveats to that. So let's talk about what a polypeptide actually is. So we are going to use our messenger RNA, and it is going to be read by special proteins in the cell, special large proteins called ribosomes, to make a polypeptide. Just like DNA, polypeptides are made up of smaller blocks that are linked together. Recall that DNA is made up of nucleotides. RNA is also made up of nucleotides. Both of them are nucleotide chains that are strung together. Polypeptides are amino acids strung together in a long chain as well. And amino acids are linked similarly to the way that we link our nucleotides in DNA. So let's talk about the structure of an amino acid. Amino acids have 4 main parts. They have a central carbon. They have an amino group, which we also call the N-terminus. That means the N end. And then we have a carboxyl, or this carbon-oxygen-oxygen group, also called the C-terminus, or the C end. All amino acids have those 3 basic parts. They differ in their R groups. So R groups can change. There are 20 different R groups. making 20 different amino acids. Each of those groups has different chemical properties, and we'll talk about that in a minute. Amino acids come from diet, so we can ingest amino acids. Some amino acids can be synthesized by the organism that needs them. Humans can't synthesize all amino acids, so we do have to get some from our diet, which is why maintaining a proper protein balance in your diet is really important. And then we can also get amino acids by recycling and breaking down other macromolecules, other proteins. So we'll talk in more detail about how we actually make polypeptides from RNA. The whole process of transcription gets its own chapter, so we'll talk about that a little bit later. This picture shows the different R groups and the different types of R groups we have. So each of the R groups gives different properties to the protein that they are a part of. These are going to be different chemical properties. And these properties affect how the proteins behave in the cell. So how they interact with water, how they interact with other proteins, and how they function. So there are five major types of R group. We have amino acid R groups that are polar, which means they have a region that is charged either positively or negatively. So in this case, the R groups have a region that is positively charged. We can also have negatively charged amino acids. So those obviously are going to interact differently with other molecules. Water is also negative and polar. And so water likes to interact with other things that are negative and nonpolar. We can also have polar and uncharged. So this means that the molecule as a whole has sort of a charged region. So in this case, we have kind of a negative-ish region in one part of the cell, but overall the molecule doesn't have a particular charge. We can also have non-polar side chains. This means no charge at all, partial or complete, no charge at all. And so those side chains are not going to interact well with water. They will interact well with things like lipids, which are non-polar as well. And so each of these types has very specific function within the protein. They can often produce regions of polar or non-polar areas that allow them to have a very particular function. But then we have a couple of one-off cases down here on the bottom that are not really related to the others. So these different R groups not only have their own different chemical properties, but they also affect the overall protein's properties and how the proteins are folded. So remember that folding those proteins is really important for overall function. So the R groups will actually interact to produce the correct folding and three-dimensional shape within a protein. So building an amino acid, no matter what the R group, is actually really similar to how we build our DNA molecule. Just like the DNA had a polarity, the 5' to 3' direction, where we always add a new DNA nucleotide to the 3' end. Amino acids and the peptides that they make up also have a direction. One end is going to be that N terminus end.
This is Kate from your American Red.
I apologize for that. I've recorded this video 5 different times, and I will not re-record it simply because Kate from American Red Cross really needs to talk to me. All right, as I was saying, amino acids and polypeptides have a direction as well. One end is the N-terminus end, because we have a free nitrogen. The other is the C-terminus, because we have a free carbon end. Now we are, just like DNA is always added at a particular location, we are also always going to add our amino acid at a specific location. We always add to the C-terminus end. So the amine group comes in, is bound to the carboxyl group, and when we do this, we create a peptide bond. So now we have a bond here between our 2 amino acids, and we can continue making a chain just like we make a DNA chain. Polypeptides can be thousands of amino acids long. And the length depends on the gene length and the mRNA length that you're using to make them. So we will talk about the specifics of how you actually read a messenger RNA and bring in the right amino acid to make a chain in chapter 13. But as a just sort of a quick overview, the messenger RNA is actually read by protein complexes called ribosomes. And those ribosomes read groups of 3 nucleotides at a time. Those groups of 3 are called codons. Remember, they are read 3 at a time. Codons are non-overlapping. That means once you start a sentence, you don't drop letters, skip letters, anything like that. And once you start, you set a particular reading frame. That means every 3 amino acids with no skips, jumps, hops, whatever, every 3 nucleotides are read for 1 amino acid. So if our top strand of DNA here is our coding strand, and we use the bottom template strand to make a complementary mRNA, we get that mRNA there that is the same as our coding strand, with the exception that we have incorporated uracil because we have RNA. Now the ribosome is going to come by and start reading its 3-letter words. 3-letter words are a good thing, not like 4-letter words in the American language. 3-letter words are going to be read as codons, and each of those codons specifies a specific amino acid. So let me show you why the reading frame is important. Let's say we have a sentence, the red cat ran. If you start that sentence in the right spot, your sentence makes sense. The red cat ran. Now we are going to keep our triplets together and make words that make sense. But if we start in a different place, we have a different reading frame. So if we start at the second letter, we have a different reading frame. Now this one doesn't make any sense to us because we were using 3-letter words, but it makes a very different sentence. In our case, it doesn't actually mean anything except for the first word, which is her. So there are actually 3 reading frames for any gene. You can start at the first letter to make the first reading frame, and if we do that, reading our codons, we get a specific amino acid chain. If we start at the second reading frame, we get a different amino acid chain. And if we start at the third, we get a third amino acid chain. So establishing the reading frame is really important in establishing the correct amino acid chain. Now, you may recall from the last video that you can actually have genes running in both directions on both strands. So let's say our top strand is our coding strand, and we're running this way, and we have reading frames 1, 2, and 3. We can actually make another set of reading frames. If we use the bottom strand instead as the coding, we can have 1, 2, 3 there. So each segment of DNA actually can make 6 reading frames. And within those reading frames, the start site and the reading frame are really important for making the correct amino acid sequence for your gene. So RNA is a really useful tool because RNA is an indication of what genes are actually being expressed. So we can look at an organism at a specific point in time under a specific set of conditions, and if we can look at the RNA that's being expressed, we can see what that cell's doing. We can see what genes are actually being used. The problem with RNA is that RNA is unstable. So RNA gets degraded very quickly within a cell. Once the cell is done with it, it takes it apart and reuses the pieces to make new RNA. And part of the reason for this is that not all genes are on at all times. Sometimes you only turn on a gene for a short amount of time, and so you don't want that RNA, when it's not being used, running around making more proteins that you don't need. So RNA is unstable, but that doesn't mean we can't get some information from it in the laboratory. One process that is used very often to study what RNA is actually being expressed is called complementary DNA, or CDNA libraries. So CDNA libraries are basically a compilation of all the expressed genes within an organism at a particular point in time. Now, you make a cDNA library by using the mRNA that the cell is expressing at a specific time. And in the laboratory, you add an enzyme called reverse transcriptase. Now, reverse transcriptase works to make a complementary copy of your RNA out of DNA. So if we were to copy this messenger RNA here, we would get a single-stranded piece of DNA. Now, DNA is much more stable, and if we can even go a step further and get it back to its double-stranded form using an enzyme called DNA polymerase, we can have a stable DNA molecule that reflects the RNA that was expressed in a cell at a particular time. So using cDNA libraries allows researchers to look at changes in gene expression over time. And this isn't all you would do. There are other steps in which you could actually visualize what DNA and determine what genes were being expressed. But just sort of as a general overview, you can sort of see how this process works. Now, you might do this to compare changes in an organism. So if you have a bacteria that is subject to particular environmental conditions, or maybe you just want to see how two strains differ, you might make a cDNA library and then use some different types of arrays and computer analysis to actually determine what you have. But it allows you to sort of compare different things, or compare before and afters. Within the process of gene expression, there's a need for regulation. So I said earlier that all of our cells contain all of the DNA that we need, whether or not it's actually being used. And what's actually being used is being controlled by regulatory mechanisms, or they're being regulated. So gene regulation is really important in controlling what is expressed in each cell at a particular place in time. And we'll come back to gene regulation a little bit when we talk about transcription. There's actually an entire chapter on it that we won't probably get to, but it is really important in allowing different cells to have different functions and to express different genes at different times, or not at all, or some all the time. So every step in the central dogma is a potential point for regulation. Now we have here, this slide is a little bit busy. We have our expanded view of the central dogma, so we've incorporated replication over here. But we have DNA and transcription. So the most common point of regulation is at what we call transcription initiation. So basically, this means that transcription never even starts if you don't need the gene. There's no reason to make a messenger RNA if you don't need the gene. So that is where the process is most often regulated. And it is regulated by special proteins, special enzymes called transcription factors. So transcription factors sort of work like an on-off switch. They bind to the gene in question, and they can either bind to block transcription, or they can bind to cause transcription. Although it is not regulated as often, messenger RNA can actually still be made and then blocked at the translation step. So this might be if the cell needs to pause for a bit and not make any more proteins for just a few minutes, or if they're getting to be too many, or it can just stop the process once you get enough. So you can regulate translation by other types of RNA called microRNA, binding to messenger RNA and blocking the process of translation. You can also affect protein processing and things like that.